Let's talk about the HIV-1 glycoprotein, which is your GP160. And let's talk about the molecular detail of this thing. A little bit about the structure, which is what's rotating up here. A little bit about the amino acids themselves, so you can see some of the motifs. A motif, M-O-T-I-F, it just means a little pattern of amino acids that happen to be conserved, like they're usually the same. Even when other parts change, they're the part that stays the same. Now, the conventions you can see up here are going to be here. Let me pause that. Rotation. There we go. The conventions, we're going to show a glycan as a set of little boxes or dots. So these are, it's telling you which kind of glycan it is and what it's hooked up to. The actual HIV glycoprotein part, your GP120 part, is going to be the pink region, sort of, that I'm circling here, kind of curled like a jelly bean. This is the trimer. Now, these things that are sticking out, the yellow and green things, those are antibodies that were put in there to hold the structure together so they could actually solve the structure. Pretend they're not there. They're not doing you any favors. Same thing at the bottom. This sort of purple and orange thing, more antibodies that are holding the bottom in place. Took people a long time to figure out how to get a decent structure of HIV envelope protein. And then the folded up sort of greenish teal colored bit down here, that is going to be GP41. Now what's missing from this structure is they're going to cut off from the MPER and the transmembrane region and all of the C-terminal tail, so no palmitylation site, anything like that. All right, so beta strands are going to be like little ribbons with a little arrow at the end of them, so you can see a couple of them right here. And this is mostly a beta stranded protein, and antibodies are super beta stranded. If you didn't know that, now you do. Look at that. These are just the sort of binding domains. It's called the FAB of an antibody. A full antibody would have two of these on either side and a big stalk that looks just like this. Same sort of fold. It's called an immunoglobulin fold. Yeah, they're good. They're good at doing the job of sticking to things. Yeah. All right, but this is not immunology class. We don't care about these antibodies. We're here for the HIV. So let's start it turning again. There we go. All right, and so you can see the 1, 2 at the back, 3 just coming around here. There's your 3 monomers, and so you got 3 GP120s surrounding 3 GP41s. You can see the 3 little alpha helices right in the center. Those are easy to count. And then this is the pre-fusion form. So the GP41's all folded up like this, and HR1 and HR2 are not yet snapped together because they can't do that yet. That's in the future. Okay, so we'll just leave this spinning up here, refer back to it if you want to. Mostly turn your attention over here, and let's look at the multiple sequence alignment. Multiple sequence alignment, it's going to be amino acids, and so they're in single letter amino acid codes, like A for alanine, C for cysteine, there is no B, yeah, amino acid, etc. If you haven't learned them, go learn them. This will make more sense after. You'll pick up something, but go learn them. Yeah, just do it. Just do it. You might as well at some point. All right. And these are colored. The program I'm using is called JALView over here, and that's a free sequence alignment viewer. You can download it and install it and view the sequence alignment all by yourself if you want to. It's what I use for work. You'll see some features here. So this is the very beginning. So we call that the amino terminus or N terminus of GP120. And there we go. We start out with a kind of a variable region. Now, you know that some of the variable regions are called variable loops, like V1, V2, V3, V4, V5. Yeah, there's 5 variable loops. Let's see all of those. This first region, this would be like V0. It's before any of the conserved or variable regions. We just pretend it's not there. Just don't comment on it. I know you can see it. Just pretend it's not there. Ugh, yeah. Gross. All right, so here we go. This is conserved region 1. And you can see in this coloring scheme, amino acids are going to show up as a color if they are conserved, like if they appear most of the time or all of the time in these sequences. The color is going to tell you what kind of amino acid you're dealing with. So if you got these sort of light kind of baby blues, those are going to be hydrophobic ones. And so those are going to want to hide from water. You're probably talking about something on the interior of the protein. You've got a couple of special ones. So the positive charged ones are going to be in red. The negative charged ones are going to be in purple. And G and P are going to basically just disrupt other structures. They're kind of marking in between alpha helices or in between beta strands. And so you can see those are always orange and yellow. Polar things like N, S, T, Q sometimes, those are going to show up as green. And some of these aromatic amino acids like H and Y and sometimes F are going to show up in this kind of teal color over here. And cysteines are special. Those are going to get their own color, which is this sort of, I don't know, it's not quite salmon pink. I don't know what you call that. It's, yeah, unearthly pink. I don't like it. Yeah, not Barbie pink for sure. All right, so there you go. That's the C1 region. You can see, yeah, that's real conserved compared to the stuff before and the stuff after. You get the little gaps in here. Those have been inserted in the alignment because some sequences have some, amino acids there and other sequences do not. And so you're just keeping all the things that are the same stacked on top of each other, and then they'll show up in colors. That's the beauty of multiple sequence alignment. All right, what is this? First region, which is variable and is in between conserved regions. Why, that would be the V1 loop. Also on the screen, can you see the V2 loop? I'll bet you can. It's real close to the V1 loop, so it's right over here. Why is it called variable? Well, you can see there's pretty much nothing lighting up in there, which means there's nothing conserved, which means, yeah, it's variable. These are things that antibodies will sometimes stick to, but because the virus can change it and it doesn't really matter to the structure, it's not important. It's a way the virus can evolve to get around any particular antibody that happens to bind these regions. So here's C2 in the middle of those two. Here's the C3 part. And in some of these conserved parts, you can see little columns of C's. C, 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 C. Those, when they're conserved cysteines, are going to be disulfide linkage sites. And so those are going to be disulfide linked. They're going to sort of hold this whole protein together. It's like a structural aid. The other thing that is worth noting in here, let's see if we can find one, are glycosylation sites. I've spotted one. So a glycosylation site for an N-linked glycan is going to have the sequence N, asparagine, something, doesn't really matter, and then either S for serine or T for threonine. And so look at this column of N, mostly V, and then T. That's a glycosylation site. And so everywhere you see that, most of those are going to end up being glycosylated. They will be on the surface of the protein. They're the spots where all these little colorful balls and squares or cubes are going to be added. All right. You got it. You got it. I knew you got it. Let's see. So here's our C3 domain, and you got your conserved cysteines, a couple of them in here. And those are going to sort of pinch together and hold this domain together. Then you got this thing. What is this thing? This is the third variable thing, so it's the V3 loop. But you can see, just looking at it, you can see this is less variable than the V1 and the V2. Those were total chaos. Some viruses completely delete them, some fill them with spam. Doesn't really matter. Yeah, variable. What you see here is this core of, it's a hydrophobic and then GPG usually, and then something like a Q or an R, which is either polar or positively charged. That's the central domain. That's the little bit that goes right down into the receptor, the fusion receptor, and is going to interact with it. So if you wanted to make a molecule that would bind to the receptor and prevent the V3 loop from binding, that's what you would be simulating, basically. Yeah, okay? Yeah, see? It's not so hard. Structural biology, not so hard. All right, then you come to the C4, V4, C5V5, and then we get to this beautiful thing. All right, so what's next? After the GP120 part, then we've got to have a cleavage. We actually have two cleavages. They are conserved. And the cleavages are going to be at furin-like enzyme cleavage sites, which means we're going to have a couple basic amino acids in a row, and then we're going to get a cleavage. Boom. Usually there'll be like a small amino acid there, and that's where your cleavage occurs. So what we're looking for are columns of basic ones, which are red, and usually you have to have 2 or 3 in a row, and look at that. KR, and then mostly R. Yeah, and then a V. So there's one cleavage site, and then RE for some reason, RR or KR, and then A, and then V. So there, boom, 1, 2. 2 cleavage sites. This is the one that sort of potentiates. This is the one that makes it easier to cleave the next site. Here's the second site that gets cleaved. Awesome. And what is right after the cleavage site? You're flipping through your notes. You know what it is. You don't need those notes. It's the fusion peptide. That's why we've got a cleave here. And this thing with these little columns of G amino acids. G is glycine. Glycine is super flexible, so that allows this thing to, it's going to form a little hook, like a little alpha helical hook. And then you see the rest of the stuff that's in there is going to be light blue, which you remembered is hydrophobic. So it's mostly hydrophobic with some flexible amino acids, and that's your fusion peptide right there. Can't argue with that. All right. So now I made some little notes so I can remember where I am. So we're past the furin, we're past the fusion peptide. Oh yeah, and then HIV doesn't mess around. Other viruses like this, they'll have a spacer domain, they'll do something fancy. No, HIV is not fancy. HIV doesn't play like that. Right after your fusion peptide, this little bit here, like SITLT, that bit is going to be the start of heptad repeat number 1. And that's the long heptad repeat. So it's going to go down to somewhere like right around here. I think that's about where it ends. And so that's going to form a long alpha helix, and the alpha helix is actually going to get bent, if I remember right, in the middle, I think at around this point, in the pre-fusion conformation. It's going to get sort of folded over a little bit unhappy. It would just like to be a nice helix. All right, then we got to scroll down a little bit, and so we're going to have another little region, and it's going to get sort of looped out. There's a cysteine. There's another cysteine. So we're going to pinch those together, or disulfide bond those together, and we're going to pull this thing to the side so it doesn't get in the way of HR1 and HR2, which are the two lovers that have to meet, like little Romeo and Juliet. Except that HR2 is tiny, so I don't know. Yeah, whatever. Big Juliet, little Romeo, little, I don't know, whatever you want. Whatever works in your mind is going to be just fine. Okay, now we got to have HR2. HR2 is itty bitty. It's going to run at about 770 in this alignment, so it's going to start right there. And yeah, that is heptad repeat number 2, just in that area. That's what's going to fold up onto heptad repeat number 1. So 1 is big, 2 is little, and clips onto the side. There you go. What's after heptad repeat 2? Pretty much immediately, you're going to have your MPER. And that's the membrane proximal, I never remember what the ER stands for, but it's the thing that's going to bind to lipid molecules, and in some other viruses it'll bind to cholesterol, but doing the same job. What you're going to get there are going to be a bunch of aromatic and or large amino acids, things like W's and Y's and F's, and here you can see W, something W, Y, that's your motif. That is going to be your MPER. Membrane proximal envelope region, something like that. I don't know. Right after that, you're going to have this mostly hydrophobic bit with, weirdly, one basic amino acid in the middle of it, just for spice, I guess. You need about 14 amino acids to get across the membrane, so it's going to stop right about here and start right about there. That is going to be the transmembrane. And then all the rest of this from about 845 onward, that is all the cytoplasmic tail. There are a couple of somewhat to mostly conserved cysteines in this region. Those are the things that are going to be palmitoylated to sort of help to anchor the bottom of the glycoprotein GP41 into the membrane. Yeah, and then the rest of this is here to interact with the other viral components, or to get out of the way, or to get transported to the right part of the cell. There's all kinds of signals just sort of stacked up in this region, and that's what some of these conserved amino acids are that are over here. All right, there you go. I think that's probably enough detail on the the envelope protein of HIV. But you got a different perspective, and I hope that makes it easier to sort of get locked in your head. And yeah, there's a lot of stuff in here that you can talk about for real. All right, thanks very much.